This is the PromethION24. So it takes 24 flow cells. So each flow cell is like that. It's a tiny flow cell. It's got a lot of pores. It can have a maximum of 12,000 pores. So the flow cell goes at the source. So this is a 24 model. So that means it can take a maximum of 24 flow cells. The flow cells go here. So you can see the slots. So you take the flow cell, push it in, then one more time, and then push it down, like that. And the PromethION system is connected to the computer, which is again from PromethION. So once you put the flow cell in the slot, so you see on the computer, it automatically on its position, it says it's ready for sequencing. It detects it and automatically it says it's ready for sequencing. To check the number of pores, you go on the start and then you click on flow cell check. So I've done it just a while ago, so I'm not doing it again. So if you can look here, so I've checked these two flow cells, 1 and 2. So I have 6,752 pores here and 3,050 because this was a used one, so that why it has less pores. For running the samples, the first thing we have to do is prime the flow cell. So at the moment I'm priming these two flow cells. So for that you prepare the priming solution and add 500 microliter of priming solution in the port, which is here, is it this one and this one. Here. So here. So I'm going to load the 500 microliter. This is the second one, and then slowly release. It's almost done. So release some liquid at the end, and similarly for the other one. So in total, we have to do the priming. We have to load the priming solution twice, 500 microliter each. So first time you load 500 microliter, then you wait for 5 minutes. Then after the 5 minutes, you load the rest of the 500 microliter. And once you're done priming, then you load your library, which I'm going to load. Okay, next we have to load the library. So here is the library. It's the cDNA library, so it comes with a base. So the total volume is 150 microliter, and I will load all the 150 microliter. The most important thing is that you should mix the library just before loading because it comes with a base, so it must be thoroughly mixed. So release a small volume to avoid any bubble. So and slowly into the flow cell. So leaving some volume at the end just to avoid any air. Just like that. And then close the lid and put the light shield. Okay, so after loading the library, you should wait at least 10 minutes. So in the meantime, you can enter some of the details. So go on the start sequencing, experiment name, and then choose the position of the flow cell, sample ID, sequencing. Continue. Select your kit. Continue. With length 2, do not find with that. Advanced options. I don't want it to scan every half an hour, so maybe 3 hours. And then click continue. And then base coloring, yes, I want the base coloring on, and I also want the modified bases. Edit options. I want both, and I want fast base calling. Apply. Continue. Data allocation. So it will save with FAST5, FASTQ, and PEM files. Filtering on. I don't want the filtering on, so I will do it later. So I will turn this off. Advanced options, nothing. So continue to final review. So I'm happy with everything, and then I will click on start. Okay, now it's sequencing. So similarly, I have to enter the detail for the other one. This is the sample. Okay, I go on the start sequencing, experiment name. Okay, so I've started both the runs, so both are running. So what I will do the post scan first, and then start sequencing. So if you can click on it, so it shows all the detail. So it hasn't sequencing yet. So once you start sequencing, we will see it's coming up. So at the moment it's not sequencing. So we don't have anything at the moment. So we have to wait a while. Okay, so this is how it looks like. So it will take some time to stabilize. So during this time, it will give fluctuated readings. How many pores are sequencing, how many pores are available and unavailable, so things like that. So it will take a while to stabilize everything. So let's go and have a look at the other one, the used flow cell. This was the new flow cell. So the used flow cell is still getting to 31.5. Okay, start sequencing. Go. Right, haven't started sequencing yet, so wouldn't take long. Because it's a used flow cell, so obviously most of the pores should be unavailable. Okay, something started to happen. Yeah, again it will take a while to stabilize. So let's go back to the other flow cell that you want to see. So it's doing post scan. So after the post scan, we'll be able to see what's going on. Okay, so it has finished the post scan, and now it's in the sequencing mode. So let's go and see. So yeah, okay. So the values are still fluctuating, and they will be fluctuating for a few minutes before it stabilizes. So at the moment we have like 700 pores sequencing, 600 available, 300 unavailable, and 1100 inactive. Go to the next screen. So here it shows minute by minute in a bar chart form. So after 4 minutes we have 28.5% sequencing, which is a bit low at the moment, but let's see how it goes. So let's go on to the other one and see. Still sequencing, still doing post scan. So if you look at the screen, it shows you the total time of running, remaining time, temperature, which should be constant. Read counts, so at the moment we have nearly 60,000 reads, and estimated basis is 47 MB, and 50 is 1.46, and here is the basis happy base called. Okay, so the used flow cell has also started sequencing. Let's go and see. Right, so 1,300 pores are inactive, 400 sequencing, 700. Again, because it's fluctuating, so we have to wait interval. So after 4 minutes, 22% pause. That's not bad actually compared to the new one, but we have to wait a few more minutes. Okay, with the new flow cell, so we are at about 90k read counts, which is quite good, and 800 pose sequencing. Oh wow, yeah, so now we're nearly 49% for the sequencing at the moment. Yeah, so this is quite good because almost all the pores which are available, they are sequencing. Okay, so we'll let this continue for 72 hours or more, and then we'll see how it goes. So thank you very much for watching.